Okay. That's good. So I'm going to start the lecture. We've met before. I'm Dr. Ari Lawal from the Department of Biochemistry, University of Lagos. Today we are going to be looking at some of these topics. We're going to introduce ourselves very quickly to metabolism and bioenergetics. We're going to talk about breakdown of biological molecules and their uses as fuel. We're going to look at biological oxidation, the electron transport chain, as well as talk about oxidative phosphorylation. Now, bioenergetics is otherwise known as biochemical thermodynamics. We know very well that thermodynamics has to do with the movement of heat around components of a system. Now, in biological molecules, the free energy is said to be the useful energy within an open system like biological molecules are. Now, the Gibbs change in free energy is that portion of the total energy change in a system that is available for doing work. So it is that energy available to carry out work. Enthalpy, on the other hand, is the heat content of the reacting system. Now, there's also another biochemical thermodynamic parameter referred to as entropy. It is the extent of disorderliness or randomness of the system and becomes maximum as equilibrium is approached. Now, these biological systems that we're talking about, living organisms, they conform to the general laws of thermodynamics. Now, we know very well that the first law states that the total energy of a system, including its surroundings, remains constant. Now, biological systems are isothermal. They don't allow heat to be conducted through them. So essentially, biological systems are able to convert energy. They do a lot of transmutation of energy from one form to the other, from chemical energy to mechanical energy and otherwise. Now, the second law of thermodynamics states that the total entropy of a system must increase if a process is to occur. Now, there's a link between the heat content of a system, the entropy, as well as the free energy that is available to carry out, to conduct work, which we are more concerned about, because we are looking at how all of this energy that comes into a system is utilized. In what form is it utilized? You can mute yourself. I have not. Please mute yourself. So essentially, the three thermodynamic parameters are related. Now, ordinarily, the free energy, when the free energy change is negative, the reaction is said to proceed spontaneously with the loss of free energy. That means it is hexagonic. Now, on the other hand, if the free energy change is positive, the reaction is not able to proceed. It can only proceed if free energy can be gained from another system. So essentially, this is an endergonic system or endergonic reaction. Now, if the magnitude of the free energy change is great, the system is stable. Now, if the free energy change is zero, that means the system is at equilibrium. No net change takes place. It doesn't, it's not able to carry out any work. Now, the cells are said to be isothermal systems. They function at essentially constant temperature. the heat flow is not a source of energy for cells, like I told you earlier, because it can do work only as it passes to a zone or object at a lower temperature. And biological systems are not programmed that way. So the energies that cells can and must use is the free energy. So it's described as Gibbs free energy function, G. In practice, an endergonic process cannot exist independently. Exergonic processes are those processes that do not readily proceed. They cannot go on spontaneously except when they are coupled to an exergonic process. So hence you have a coupled exergonic endergonic system. Those are the only systems that can allow an endergonic process to occur readily. Now, what are these exergonic reactions? Exergonic reactions that will occur readily are termed catabolisms. Generally, the breakdown as well as the oxidation of fuel molecules are referred to as what? As catabolism. However, synthetic reactions that build up substances, those are the energy requiring processes. They are referred to as what? Anabolism. Now, the sum total, the total of all of these catabolic and anabolic processes that occur within living system is referred to as metabolism. This pictorial indicates where an exergonic reaction is coupled to an endergonic reaction. So like I said earlier, endergonic reactions like synthesis reactions will not occur except when they get energy from an exergonic reaction. Now, what are high energy phosphates? The high energy phosphates are known to play a central role in energy capture and transfer. What do I mean? ATP, which is the universal currency of energy, is a high energy phosphate, even though it is not that molecule with the highest free energy change on hydrolysis, but it is a high energy phosphate. It has some characteristics that we'll talk about shortly. Now, these high energy phosphates, because when they are hydrolyzed on hydrolysis, they yield a high free energy change on hydrolysis. Example of such high energy compounds: phosphoenol pyruvate, carbamoyl phosphate, phosphoglycerate, creatine phosphates, glucose 1 phosphate, ATP, ADP. They are all high energy phosphate groups. Now, these high energy phosphate groups, ATP is an intermediate.
1 hour. 4. Give it to
Now, the ATP, like I said earlier, ATP has the high phosphoric transfer potential. Apart from being a high energy phosphates group, it has a high phosphoric transfer potential due to the negatively charged phosphate groups, or the negatively charged oxygen attached to the phosphate groups in the phosphoric acid component of the ATP. So essentially, ATP on hydrolysis yields a high free energy change, which you can couple to an endergonic process to drive the endergonic process. But not only that, it also has a high phosphoric transfer potential. That means its ability to quickly donate its phosphate group would hence make it a good material as a high energy phosphate compound, or the energy currency of the cell. Now, metabolism. I said earlier that metabolism is the sum total of chemical transformations taking place in the cell or organism, and they occur through a series of enzyme catalyzed reactions. We know very well that within the cell, what drives a lot of processes, both the anabolic and catabolic processes, are enzymes, which are proteins or some other catalytic molecules. Now, this series of enzyme catalyzed reactions constitute what you call the metabolic pathways. Now, each of the consecutive steps in a metabolic pathway brings about a specific or small chemical change. This change is usually the removal, transfer, or addition of a particular atom or functional group. Now, the precursor is converted into a product. So most times we talk about a substrate and a product. So the substrate is converted into a product via the activity of the enzyme. So it's converted into a product through a series of metabolic intermediates. These metabolic intermediates are referred to as metabolites. So if you have a substrate, if a substrate is producing a final product, in between those metabolic intermediates before it gets to the final product are referred to as metabolites. Now, the two phases of metabolism. The first one is catabolism. Catabolism is degradative. That's the degradative phase of metabolism, in which organic nutrient molecules such as carbon, fats, proteins, nucleic acids are converted into smaller and simpler end products such as lactic acid, carbon dioxide, ammonia, and others. Now, catabolic pathways release energy. You remember very well that hexagonic reactions will take place with a concomitant production of free energy that can be used to carry out work. Now, catabolic pathways release energy. Some of this energy will be conserved in the formation of ATP, or some of this energy will be used or conserved in generating ATP, while others are conserved in reduced electron carriers, which is very central to metabolism. These reduced electron carriers are NADPH, NADPH, and FADH. That's reduced nicotinamide adenine dinucleotide, reduced nicotinamide adenine dinucleotide phosphate, as well as flavin adenine dinucleotide. Flavin adenine dinucleotide. These are reduced forms of these organic compounds. Now, the catabolic pathways are degradative. They are also hexagonic in nature. They release energy, and some of this energy is most times captured in synthesizing other or in driving other processes. Now, anabolism, which is the biosynthetic phase in which small simple precursors are built up into larger and more complex molecules, whereby you have fatty acids and glycerol forming lipids, glucose, as well as some other monosaccharides like mannose and the rest forming polysaccharides, amino acids forming proteins, and then nucleotides forming nucleic acids. These are biosynthetic reactions, and they are most times endergonic in nature. They require an input of energy, generally in the form of ATP or reducing equivalents, which can drive these anabolic processes. So anabolism and catabolism, these are some of the differences between them. The starting reactions are different. In anabolism, the precursor in anabolisms are amino acids, sugars, fatty acids, nitrogenous bases, and smaller compounds. So the precursors, on the other hand, in catabolism are energy-containing nutrients, which we refer to as fuel molecules. Carbohydrates, fats, proteins, they are fuel molecules. Those are the starting materials in catabolism. So when they are broken down, the final product in catabolism will be energy-depleted end products, such as carbon dioxide, water, and ammonia, while the end product in anabolic processes are proteins, polysaccharides, lipids, and nucleic acids. Now, in anabolism requires reducing equivalents to drive them. Reducing equivalents such as FADH, NADPH, and NADH, while catabolism generates those reducing equivalents such as NADH, NADPH, and FADH. On the other hand, too, ATP is required for anabolic reactions. ATP, as we already know, are energy conserving molecules or energy currency. So they also help to drive anabolic processes via the hydrolysis. So hydrolysis of ATP would yield a high free energy change that can be used to drive anabolic processes. While on the other hand, catabolism requires energy depleted currencies such as ADP and oxidized NADH, FAD, and NADP plus. So these are the molecules that are required for catabolism to take place, because catabolism will now produce ATP as well as the reduced organic materials like NADH, NADPH, and FADH. Now, the strategy of metabolism, the first strategy of metabolism, or the first condition for which metabolism takes place, is one, to generate ATP. The second is also to generate this NADPH or NADH, FADH, which are reducing equivalents. The third is that metabolism occurs to synthesize and degrade biomolecules that are required in specialized cellular functions. The lipids are degraded into acetyl-CoA. Molecules are synthesized, glycogen and the rest of them that form part of the muscles are synthesized via these metabolic processes. Now, the fourth strategy is also to alternate degradative and biosynthetic pathways. Now, if synthesis is occurring, and then when synthesis occurs, whatever is generated is degraded too, that means you will have a futile cycle. It is not regulated. So metabolism exists to regulate these degradative and biosynthetic pathways, so that we won't end up with a futile cycle. Now, these are some of the metabolic pathways that we have. There are some pathways that are linear. Example of a very linear pathway is the glycolytic pathway, while others are non-linear. They are either branched, converging or diverging, or cyclic, like you have in the tricarboxylic acid cycle. So some of them are linear, like the glycolytic pathway, while others are non-linear. Now, these are examples of converging metabolism, where glucose, starch, and sucrose can produce glucose. The glucose and the rest of all the other fuel molecules, such as fatty acids, amino acids, they can all converge in the synthesis of acetyl-CoA. Now, the acetyl-CoA so produced can now go into the cyclic tricarboxylic acid cycle to generate reducing equivalents and GTP, while some other molecules too can produce several other intermediates. One molecule produce 3 intermediates. That's the diverging anabolism. So these are some of the pathways we have. Overview of metabolism. The fuel molecules, carbohydrates, proteins, and fats, they are digested and absorbed within living cells. Carbohydrate is broken down into simple sugars. The simple sugars can be interconverted into glucose, all of the other monosaccharides that can be transmitted into glucose, while proteins, when they are broken down and absorbed, are absorbed in form of amino acids. Fats, on the other hand, will generate fatty acids and glycerol, which all of these simpler molecules, simple sugars, amino acids, and fatty acids and glycerol can be broken down further into acetyl-CoA. The acetyl-CoA then passes in to the final oxidative, final common pathway for the oxidation of fuel molecules, which is the tricarboxylic or citric acid cycle, to generate carbon dioxide and reducing equivalents, which will produce ATP in the long run. Now, these are some of the interconversion of monosaccharide, mannose, produced from any other carbohydrates, can be converted into fructose and then into glucose, as shown here. Galactose, on the other hand, can also be converted back into glucose, as we can see here, galactose to glucose. Then fructose too can also form some intermediates of the glycolytic pathway and then go back to produce glucose. Now, we know very well that glucose will be the starting point of the oxidation of sugar within the body system. Now, the first main pathway, glycolysis. Glycolysis is said to be the major pathway for glucose metabolism. Now, there are two things that could happen to glucose. Glucose can be broken down via the glycolytic pathway or the pentose phosphate pathway. Glucose can also be stored as starch in plants or as sucrose or glycogen in animals. So glucose can be stored or can be broken down. The glycolytic pathway is the major pathway for glucose catabolism. And this pathway, we're talking about the features of this pathway now. This pathway shows that glucose is the major fuel for most organisms. Almost all organisms have are able to carry out the glycolytic pathway. So the glycolysis in itself is an anaerobic process. It doesn't require oxygen. So it consists of about 10 enzyme catalyzed reactions. It consists of about 10, nearly every living cells are able to catabolize glucose and other simple sugars by this process called glycolysis. It differs from one species to another only in the details of the regulation as well as the fate of the pyruvate generated in this glycolytic pathway. Now, glycolysis is the metabolic pathway that catabolizes glucose into 2 molecules of pyruvate. It is a 10-stepped process. It consists of one, the preparatory phase and the payoff phase. What we call the preparatory phase is that phase where ATP is utilized. 2 molecules of ATP is utilized, 4 molecules of ATP is generated in the other phase, which is the payoff phase. So glycolysis, like I said, consists of 10 enzyme catalyzed reactions. One major feature of glycolysis is that all of its intermediates are phosphorylated. All of the intermediates are phosphorylated. The pathway can be broken down into 2 phases, like I said. Now, the next reaction in glycolysis produces 2 molecules of reduced nicotinamide adenine dinucleotide, 2 molecules of ATP, as well as 2 molecules of pyruvate for every mole of glucose broken down. So this is the next reaction involved in the glycolytic pathway. This is the schematic. There are enzymes. That lead to the final production of pyruvate. So this is the preparatory phase. The preparatory phase ends with the production of glyceraldehyde and dihydroxyacetone phosphate, as you would see in your notes, while the payoff phase, this is glyceraldehyde and dihydroxyacetone phosphate. The dihydroxyacetone phosphate can isomerize into glyceraldehyde 3-phosphate. So hence, moving into the payoff phase, 2 molecules of glyceraldehyde 3-phosphate goes into this payoff phase. Now, the payoff phase leads to the generation of 1 mole of pyruvate. I talked about the energetics earlier. There are 2 steps where ATP is utilized, and there are 4 points where ATP is generated. So the net ATP generated in the glycolytic pathway is 2 moles of ATP. Now, one very important thing about the glycolytic pathway is its efficiency. The efficiency is how much it is converting the ATP that it uses into more ATP. It is only able to generate just 2 moles of ATP. So if it generates 2 moles of ATP, its efficiency is just about 2.96%. That means there are still more ATP that can be generated if the pyruvate generated in glycolytic pathway can be broken down further in the presence of oxygen. So remember that the efficiency of the glycolytic pathway depends on the availability of oxygen, and oxygen is not used in the glycolytic pathway from glucose to pyruvate. So moving further, the pyruvate needs to be broken down further to generate more ATP that is still trapped within those hydrocarbon bonds. Now, the inhibitors of the glycolytic pathway, what are the inhibitors of the glycolytic pathway? Two major ones here. Aldoacetate is known to inhibit the glyceraldehyde 3-phosphate dehydrogenase step. That means it's able to inhibit an enzyme within the glycolytic pathway. Fluoride that is contained in most of these mouthwash also inhibits the enolase step. It prevents the enolase from acting such that the glycolytic pathway will not be able to generate the pyruvate. So what are the fates of pyruvate? The fates of pyruvate, there are two conditions. Under aerobic conditions, as you see in your notes and the textbook, under aerobic conditions, there's a pathway for pyruvate, and under anaerobic conditions too, there are also pathways for pyruvate. Now, under aerobic conditions, that means the pathway, the major pathway for the degradation of pyruvate is the oxidative decarboxylation of pyruvate into acetyl-CoA. Now, oxidative decarboxylation of pyruvate into acetyl-CoA, as the name implies, involves oxidation as well as decarboxylation of this pyruvate. And the enzyme involved, the enzyme that catalyzes this reaction, is a complex enzyme consisting of three enzyme components. It's referred to as the pyruvate dehydrogenase complex. It has three enzyme components. One is the pyruvate dehydrogenase component, the dihydrodepoyl transacetylase, and then the dihydrodepoyl dehydrogenase. So in the oxidative decarboxylation of pyruvate, apart from generating acetyl-CoA as shown here, it also generates NADH. So for every mole of pyruvate that goes through oxidative decarboxylation, 1 mole of acetyl-CoA and 1 mole of reduced nicotinamide adenine dinucleotide here is produced. Now, under anaerobic conditions, pyruvate can yield ethanol, can also yield lactate via the activity of pyruvate decarboxylase or lactate dehydrogenase as shown here. Okay. Now, also, pyruvate can also be converted into the amino acid alanine via the activity of an aminotransferase enzyme. So it involves transamination, whereby the pyruvate, which is a keto compound, takes up an amino group from another amino acid and then produces alanine and then a keto, another keto acid from the old amino acid. So you have an alanine aminotransferase enzyme carrying out this transamination reaction. Now, pyruvate, like I said earlier, can be converted into lactate as shown here. The enzyme is lactate dehydrogenase, and anytime dehydrogenases normally have NAD linked. That means they have the nicotinamide adenine dinucleotide with them, assisting them with the hydrogen, either the removal of hydrogen or addition of hydrogen. So lactate dehydrogenase can interconvert between pyruvate and lactate, while conversion of pyruvate to ethanol involves a two-step reaction. One is conversion of the pyruvate into acetaldehyde, and then the reduction of the acetaldehyde by alcohol dehydrogenase into ethanol. The tricarboxylic acid cycle, which is the TCA cycle, is the final common pathway for oxidation of fuel molecules such as carbohydrate, lipid, and protein. The tricarboxylic acid cycle is also a series of enzyme-catalyzed chemical reactions, and it forms a key part of aerobic respiration in cells. Glucose, fatty acids, and most amino acids are metabolized to acetyl-CoA or intermediate of the cycle. Now, this cycle is also referred to as the Krebs cycle or of the citric acid cycle. The process takes place in the matrix of the mitochondria. This is the pictorial of the citric acid cycle, with acetyl-CoA being the main precursor. So acetyl-CoA goes in, and in the citric acid cycle, there are points where NADH as well as FADH are generated, and then there is also a point where ATP or GTP is generated here, the succinate thiokinase step. So there are about 3 points where NADH, which is a reducing equivalent, is generated. Now, this reducing equivalent, they are potential sources of energy. That means they can produce ATP if they are taken to the electron transport chain, which is a series of enzymes on the inner mitochondrial membrane. So in the electron transport chain, the NADH, the reduced nicotinamide adenine dinucleotide, are further oxidized to generate more high energy phosphate groups like ATP. So hence, the citric acid cycle, the energetics of the TCA cycle. In the TCA cycle, more ATP can be generated via, so the TCA cycle produces 1 mole of FADH as well as 3 moles of NADH, and then it produces 1 GTP, which is equivalent to 1 ATP. Now, the 3 moles of NADH, if they go into the electron transport chain, they can be oxidized into 9 ATP. The 1 mole of FADH, every 1 mole of FADH is oxidized within the electron transport chain and can yield 2 ATP via oxidative phosphorylation. Now, hence, in a cycle of the tricarboxylic acid cycle, 12 moles of ATP can be generated per cycle. Now, since we know that 1 mole of glucose generates 2 moles of acetyl-CoA, which can form 2 moles of pyruvate, which are formed from 2 moles of pyruvate. Hence, 2 cycles of the TCA cycle will generate 24 moles of ATP. Now, oxidative decarboxylation of 2 pyruvate to acetyl-CoA yields 2 NADH, which is 6 ATP. So if we add that to the 24 moles of ATP generated in the 2 cycles of TCA cycle, that means oxidation of pyruvate in the mitochondria will yield approximately 30 ATP. These are subject to some changes based on the transporter molecules. Now, the regulation of the citric acid cycles. Low level of ATP will stimulate the formation of acetyl-CoA from citric acid cycle. For the citric acid cycle. Now, high ATP and NADH levels will decrease the formation of acetyl-CoA and it will definitely slow down the citric acid cycle. It will send a signal back that there should be a stoppage to the synthesis of acetyl-CoA. So hence, slowing down the citric acid cycle, since there is a high amount of ATP as shown here and high levels of NADH. So high energy level. So high energy level would slow down the TCA cycle. The pentose phosphate pathway, on the other hand, is an alternate route for the oxidation of glucose without direct consumption or generation of ATP. So the essence of the pentose phosphate pathway, it takes place within the cytosol too, because the NADP plus is used as an hydrogen acceptor. It is also known as the exose monophosphate shunt or the phosphogluconate pathway. It is basically used for the synthesis of NADPH, that's the reduced form of nicotinamide adenine dinucleotide phosphate, as well as synthesis of the ribose component of nucleic acids. Now, the pentose phosphate pathway has 2 phases. One is the oxidative phase, where NADPH is generated and decarboxylation occurs. There are 2 moles of NAD for each mole of glucose oxidized in the pentose phosphate pathway. 2 moles of NADPH is generated, and then the non-oxidative phase. In the non-oxidative phase, the ribulose 5-phosphate generated in the oxidative phase is now broken down or rearranged. The carbons in the ribulose 5-phosphates are rearranged to form other useful sugars like fructose 6-phosphate and the erythrose 4-phosphate. Now, the ribulose can also be converted or isomerized into ribose 5-phosphate. Ribose 5-phosphate is a component or is a material or an intermediate in the synthesis of nucleotides such as RNA, ribonucleic acid, and deoxyribonucleic acid, DNA. So these are the uses. Meanwhile, at the end of this rearrangement, the fructose as well as the fructose 6-phosphate generated in the non-oxidative phase can go back to produce more glucose that can be taken back into the oxidative phase to produce more NADPH and then more ribose 5-phosphates. This is the oxidative phase, the mechanism of the oxidative phase, the isomerization of ribulose into ribose 5-phosphate, which is a component material required in the synthesis of nucleic acids. And then the ribulose is also epimerized into xylulose 5-phosphate, which can be rearranged in the non-oxidative phase into pseudoheptulose and glyceraldehyde, and invariably converted into glucose 6-phosphate, which can now return back to the starting point of the pentose phosphate pathway. So these are some of the rearrangements that occur. The 5-carbon ribulose, 2 5-carbon ribulose 5-phosphate can yield 7-carbon pseudoheptulose and 3-carbon glyceraldehyde. Now, the regulation of the pentose phosphate pathway. The pentose phosphate pathway is regulated via the enzyme involved in the committed step, which is the glucose 6-phosphate dehydrogenase. So essentially, when as NADPH is utilized in the reductive synthetic pathways, the increasing concentration of NADP plus or the oxidized NADP stimulates the pentose phosphate pathway so as to replenish NADPH. Now, what are the differences between the glycolytic pathway and the pentose phosphate pathway? The glycolysis P is used, and it is also generated in the pentose phosphate pathway. No ATP is used, no ATP is generated. In glycolysis, carbon dioxide is not generated, while in the pentose phosphate pathway, carbon dioxide is generated in the oxidative phase. Reducing equivalents required in the glycolytic pathway, or the oxidized reducing equivalents, are NAD plus for glycolysis, while it is NADP plus for pentose phosphate pathway. So the reducing equivalent that will be generated are going to be oxidized form of NAD, and in the glycolytic pathway, while the pentose phosphate pathway, you have the oxidized form of the NADP plus. Now, in gluconeogenesis, gluconeogenesis simply means the synthesis of glucose from non-carbohydrate precursors. And what this means is that glucose can be synthesized from the breakdown of fatty acids. Fatty acids can be broken down into acetyl-CoA, which can go into the tricarboxylic acid cycle. The malate produced in the malate, which is an intermediate in the citric acid cycle or the tricarboxylic acid cycle, can now go back to produce phosphoryl pyruvate. The reactions can be a reverse or almost a reverse of the glycolytic pathway, whereby phosphoryl pyruvate goes back to form 2-phosphoglycerates. 2-phosphoglycerates forms 3-phosphoglycerate and then ultimately produces glucose, as can be shown here via the activity of some of these gluconeogenic enzymes. So these are the gluconeogenic enzymes here in color. Fructose 1,6 bisphosphatase, gluconeogenic enzymes. These are enzymes specific to gluconeogenesis. So this is the final product, the glucose. So glucose can be produced from non-carbohydrate precursors such as the lipids, or from fatty acids, could be produced from amino acids, which can also go into the TCA cycle via some of the intermediates of the TCA cycle to produce glucose. So this is a breakdown of what gluconeogenesis entails. Now, in glycogen synthesis simply means synthesis of glycogen from glucose, while glycogenolysis means the breakdown of glycogen to produce glucose. When there is a depleted level, or when there is a low level of glucose within the body cells, glycogen are broken down into glucose. While when there is excess glucose within body cells, when there is excess glucose within the body cells, the glucose is converted into glycogen or stored as glycogen. So there are also control points. There are enzymes involved in these steps too. The electron transport chain. The electron transport chain is simply a series of reactions within the inner mitochondrial membrane, where electrons are transferred from the electron carriers or the reducing equivalent NADH as well as FADH. These electrons are carried from NADH and FADH down to the final electron acceptor, which is oxygen. So the essence of the electron transport chain is one to pass on the electron to the final electron acceptor, which is oxygen. The second thing is to create within the matrix of the mitochondria a kind of hydrogen gradient. The hydrogen obtained from this oxidation of this reducing equivalents NADH and FADH generates a kind of hydrogen gradient. This hydrogen gradient can now, the hydrogen gradient now allow the hydrogen gradient is set up by complexes of proteins lining this electron transport chain. And when the hydrogen gradient is set up, it can now drive the phosphorylation of ADP and inorganic phosphate to produce ATP. So in the electron transport chain, the reducing equivalents are oxidized, and then ATP is also synthesized. So as a result of this oxidation of reducing equivalents within the electron transport chain, ATP can then be synthesized. So this is what the processes look like. NADH, and if NADH comes into the electron transport chain, there are enzymes that would catalyze the oxidation of NADH into NAD with hydrogen passed on to another molecule, an electron acceptor. Now, at the tail end of this, the electrons are passed on down to oxygen. Now, that goes on with the production of water. The NADH as well as FADH are being oxidized within the electron transport chain. Hydrogen or protons are generated within the matrix, and these protons are pumped out via some of these complex 1, 3, and 4. when the protons are pumped out into the intermembrane spaces, the space between the inner mitochondrial membrane and the outer mitochondrial membrane, the ATPase enzyme now helps to bring in some of this hydrogen. As ATPase brings in this hydrogen, it allows for the coupling of oxidation to phosphorylation. That means as oxidation is taking place, it also drives phosphorylation. So we say oxidative phosphorylation, whereby ADP and inorganic phosphates are combined to yield ATP within the mitochondria matrix. So this is so NADH and FADH are oxidized by the electron transport through the respiratory chain. The electron transport chain is also referred to as the respiratory chain. So there is a synthesis of ATP. The energy liberated by the electron transport is used to create a proton gradient. I told you earlier that a proton gradient or hydrogen gradient is set up across the mitochondrial inner membrane, and then this is used to drive ATP synthesis. And driving ATP synthesis, as it drives ATP synthesis, ATP synthesizes ATP, and ATP synthesis is also an inner mitochondrial membrane protein too. It's also found lining the inner mitochondrial membrane. So this is a pictorial of what happens when NADH and FADH are oxidized. If they are oxidized, simply NADH will yield NAD, as well as hydrogen. The hydrogens will accumulate here. This is where acetyl-CoA, we know very well that citric acid cycle produces this NADH and FADH that we can visualize here. And then the hydrogen can now be pumped out through complex 1, 3, and 4, as shown here. So it is pumped out into the intermembrane space. And at the tail end of the process, ATPase enzyme, which is not shown here, ATPase can now drive the synthesis of ATP from ADP and inorganic phosphates that are lining this matrix here. So essentially, what drives ATP synthesis in the electron transport chain here is that hydrogen gradient set up by these proton pumps, complex 1, complex 3, and complex 4. Complex 2 is the point where FADH goes in. It is not involved with pumping hydrogen. Hence, NADH can produce 3 ATP, can drive the synthesis of 3 ATP within the electron transport chain, while FADH can only drive the synthesis of 2 ATP within the electron transport chain here. So I want to thank you for listening.